Hello all, in this video we will be learning about tissue engineering. Tissue engineering usually involves the process of cell isolation, cell expansion, 3D scaffold, tissue development, growth factor of the cells and also the implantation. Tissue engineering and regenerative medicine have become largely interchangeable as the fields hopes to focus on cures instead of treatments for complex, often chronic diseases. Tissue engineering evolved from the field of biomaterials development and refers to the practice of combining the scaffolds, cells, and biologically active molecules into functional tissues. The goal of tissue engineering is to assemble functional constructs that restores, maintain, and also to improve the damaged tissues and the whole organs. Usually, artificial skin and cartilage are examples of engineered tissues that have been approved by the FDA. However, currently used However, currently they have limited use in human patients also. Regenerative medicine is a broad field that includes tissue engineering but also incorporates research on self-healing, where the body uses its own system, sometimes with the help of foreign biological material, to recreate cells and rebuild the tissues and the organs. Cells are the building blocks of tissues, and tissues are the basic units of function in the body. Generally, groups of cells make and secrete their own support structures called extracellular matrix. This matrix or scaffold does This matrix or scaffold does more than just support the cells. It also acts as a relay station for various signaling molecules. Thus, usually cells receive messages from many sources that become available from the local environment. Each signal can start a chain of responses that determine what happens to the cells. Tissue engineering and regenerative medicine work is the process of using bioengineering heart, liver, lungs, and kidney tissues. Tissue engineering and regenerative medicine work is the process of making use of bioengineered heart, liver, lungs, and the kidney. Individual cells respond to signals, interact with their environment, and organize into tissues and organisms. Researchers have been able to manipulate these processes to mend damaged tissues or even create new ones. Another method to create new tissue uses an existing scaffold. The cells of a donor organ are striped and the remaining collagen scaffold is used to grow new tissues. A biomaterial which is made from pigs' intestines, which can be used to heal the wounds in humans. Tissue engineering plays a small role in patient treatments. Supplemental bladders, small arteries, skin grafts, cartilage, and even a small trachea have been implemented in patients, but the procedures are still experimental and quite costly. Research supported by NIBIB includes development of new scaffold materials and new tools to fabricate, image, monitor, and preserve the engineered tissues. Some examples of research in the area are controlling the stem cells through their environment. For many years, scientists have searched the ways to control how stem cells develop into other cell types. I hope you would have found the video to be useful. See you in the next video. Until then, it's bye from Radhika.